Thank you, Chairman, for providing such a great opportunity to present my work. Good morning, everyone. There is nothing to disclose. Previously, we have shown that implantation of single cells derived from adipose mesenchymal cells into cryo-engineered urethra of rabbits reconstructed the functional urethra. However, there are limitations of implantation of single cells, such as there is low retention and survival of cells post-implantation. Cells could be damaged during harvesting procedure and further damaged by needles during implantation. Therefore, to overcome these difficulties, now we are using a novel technique called Kenjan system, which uses a bio-3D printer. Biofabrication of adipose-derived C-type cell structure. Adipose tissues are harvested from rabbits or cultured and subcultured. After the culture, the cells were harvested and labeled with PKS26, which gives red color to the cell and helps in cell tracking post-implantation. Then these cells were seeded into the 96 oil plates to form spheroids. These spheroids are picked up by the Kenjan system with pre-designed data to form 3D structures. And the formed 3D structures were kept into the perfusion culture for 1 week. And this is the image of biofabricated C-type AMC structure after removal from this Kenjan. Now moving to implantation of C-type AMC structure into cryo-engineered urethra. This is the image of intact urethra, which was sprayed with liquid nitrogen for 20 seconds. And a small incision within the cryo-engineered area was created and immediately implanted this C-type AMC structure, and the incision was closed. For control groups, SAM operations were performed in the similar manner. Finally, after 4 weeks of implantation, the urethras were harvested for histological and immunohistochemical analysis. Now moving to reconstruction of muscle layers 4 weeks post-implantation. These are the gross images of SAM-operated controls and C-type AMC structure implanted group. So here in SAM-operated control, as shown by arrows, you can see the urethra seems to be narrowed. However, in case of C-type AMC structure implanted group, the urethra seems to be wider and straight. This circle represents the cryo-engineered area where we performed the histology and found that the muscle layers were disorganized. However, in C-type AMC structure implanted area, the muscle was regenerated as shown by asterisks. Now moving to differentiation. These are the immunofluorescence images stained for myoglobin, SMA, S100, VWF. So as shown by arrows, most of the cells within the structures were found double positive for myoglobin, SMA, S100, and VWF, which suggests that implanted cells differentiated into skeletal muscle cells, smooth muscle cells, nerve cells, and vasculoendothelial cells, respectively. Furthermore, some other cells within the structures were found double positive for VEGF, NGF, TGF-beta1, TNF-alpha, suggesting that implanted cells secrete growth factors and cytokines. In conclusion, C-type AMC structures were biofabricated by using 3D bioprinter. The cells within the AMC structure survived for 4 weeks post-implantation and formed layered muscle structures at the surrounding neighborhood of the implanted regions. The cells within the AMC structures differentiated into muscles, nerves, and endothelial cells and further secretes growth factors and cytokines, which might be one of the reasons for regenerating cryo-engineered urethra. Thank you very much.
Thank you. Any questions? So should we move on?
Okay, thank you very much. As we're going to move on, we're running a little late. Thank you.